my name is Megan. I'm an instructor at the DNA Learning Center. Welcome to DNA LC Live. Thank you so much for joining us. Welcome back if you've been joining us these past few weeks as we've been going live. Today, we are going to be doing an extraction. Now, if you've been tuning in with us the past few weeks, you've seen Mr. Paul do a few extractions already. You've seen him do an extraction from wheat germ. You've seen him extract DNA from his own cells, which is really cool. And then today we're going to do another extraction, this time from fruit. Specifically, we're going to be using strawberries. So this is something that you can do with materials that you can find in your kitchen or around your house. So hopefully you'll be able to join me in doing this experiment. All right, but before we do the hands-on portion, we're just going to share my screen with you really quickly. All right, and hopefully you should be good and seeing this. So I'm going to start off today by telling you a little bit about the DNA Learning Center. Some of you who are tuning in may not know too much about what we are, but this is actually the building where I work, and this is the DNA Learning Center in Cold Spring Harbor. It actually was built in the 1920s and was used as a schoolhouse by the district in the area. And then in the 1980s, it was bought by Cold Spring Harbor Laboratory, and it was converted into what it's used for today. So we are made up of 4 science labs. We have a museum exhibit, which you may have seen on some of our other live videos. We also have a computer lab and an auditorium. So it's a really great place for people to come visit on field trips and do hands-on experiments that they maybe wouldn't be able to do at school. So we have all the equipment for them to be able to do it here. So as I did say, we are part of Cold Spring Harbor Laboratory, which is just a little bit down the road. And that's what you would see if you were driving by. Now, I always ask my students, if you were to pass this, would you necessarily think science lab? Probably not. I actually used to, I did an internship for the Department of Environmental Conservation, and we were down here in this harbor on a boat. And I used to look up at this area, and we used to joke around and say it was the Harry Potter houses. It looked like Hogwarts, especially because of these buildings up here. But now I work for them, which is really cool. But when I ask my students, what would you think? It's not going to be science lab automatically. A lot of students try to say that it looks like a village because of all of these houses. And then they also say it kind of looks like a college campus. And I absolutely agree with those opinions. It is kind of set up like a college campus, which is pretty unique. There are places for us to do our work, but there's also places for us to study. We have a library, there's places to eat, and there are people who live on the property as well. It is also set up a little bit like a village because it is made up of old homes. Some of these buildings are over 100 years old. This one right here is actually called Jones, and that's the oldest biology lab in the country. So that's pretty cool as well. So there are these old houses, but on the inside, they've been converted for modern science research. And the basis of what is studied over there is going to be DNA, what we've been talking about for the past few weeks with you. So in order to study DNA, you really have to have a good understanding of the shape of DNA. All right, so shape is important in things like that, but it's also important in very small things in your life. I always give the example of if you were to have a bowl of soup, you're going to go get silverware from the drawer. What are you going to take out? To eat soup, are you going to take out a fork? No, that's the wrong shape for what you're trying to eat. You're going to take out a spoon. So if shape is important in little decisions in your life like that, of course it's going to be important when we're trying to understand DNA. So let's talk about the discovery of the shape of DNA. So we have a few scientists here. We have Dr. Watson and Dr. Crick. They were 2 of the scientists who helped discover the shape of DNA. The other 2 that we mentioned are Rosalind Franklin and Maurice Wilkins here. And what happened here is that Rosalind Franklin was in the lab and she was taking photos of cow DNA using x-rays. So if you've ever gotten an x-ray before, whether it was of a bone or if you've gone to the dentist and they've taken an x-ray of your teeth, you know that you get a picture back. Well, Rosalind Franklin was getting pictures back, and this is the picture that you see here. So when this picture was shared with the other scientists, including Watson and Crick, it was almost like the missing puzzle piece. They had finally discovered what that shape of DNA was, and that was the double helix that you can see here. So some people like to describe it as a twisted ladder or a spiral staircase, and you're correct in saying that, but the term that we use is the double helix. Double means two, okay? So we have the two backbones of DNA here, made up of sugar phosphates, and then helix refers to its spiral shape. Okay, now today what we're going to be doing is we're going to be doing a DNA extraction. Well, let's think about that for a moment. What does it mean to extract? Okay, if you've ever gone to the dentist, back to the dentist, if you've ever gone to the dentist before and had teeth extracted, what does that mean? They've been taken out, right? So we are going to be taking DNA out of something today. Now let's think, okay? We know that we're taking DNA out of a strawberry, but on a microscopic level, where are we taking that DNA out of? We're going to take the DNA out of the cells. That's where DNA is kept. And specifically within the cell, it's kept in the nucleus. Maybe you've heard the nucleus referred to as the brain of the cell. Well, that's because it contains the DNA. And the DNA is basically the instructions on how to make that living thing. So I have my own set of DNA that's going to be the instruction on how to make me. The strawberry is going to have its own DNA with instructions on how to make that strawberry. Okay, remember, all living things have DNA. A lot of people get confused. They think, but I eat strawberries. Does that mean that I eat DNA? Yes, absolutely. Okay, so hopefully let's get a look at it. Now, I'm just going to stop sharing for just a moment. Now let's just think about that for a moment. We are going to be seeing DNA. That may sound a little weird to you because we're used to knowing that DNA is extremely, extremely thin. We know that it's a very long strand, but if I were to hold it up, one single strand of DNA, would you be able to see it? No, you wouldn't. What about if I put it under a compound microscope, maybe the type of microscope you have in your classroom? Would I then be able to see that double helix shape? No, you still wouldn't be able to see it. That's how truly thin and small DNA is. But the way we are going to do our experiment today is not only are you going to extract the DNA, but you're going to be able to see it with your own two eyes. No microscope necessary. All right, now we can go ahead and kind of get started with our hands-on portion. All right, so I'm just going to adjust my camera for you. All right. Hopefully you can see some of our materials there, and I'm going to come around. All right, so we have some different things that I found around my house. So as we said, we're doing an extraction from strawberries, so I had to go out and grab some strawberries. I have some dish soap. We're running a little bit low, but that's more than enough to do this experiment. I have salt. I have a few cups. Droppers, if you have them lying around, but it's not necessary. I'm probably not going to use them today, but just in case you have them around, a cup, some measuring cups and measuring spoons, cheesecloth, okay? And if you don't have cheesecloth, there's other things you can use. You can use a coffee filter, or you can even use a strainer, one of those mesh strainers. You're also going to need a plastic bag, and you're going to need ethanol, which I currently have in my freezer right now. We're keeping it chilled. All right, so let's get started. So the first thing that we're going to need to do in order to extract the DNA is we are going to have to create a soap buffer. All right, so the first part of the soap buffer, which I'm going to make in this glass, is going to be 1/3 cup of water. All right, so I'm going to add that here. Now I'm going to add 2 teaspoons of soap. All right, 1 and 2. And then I'm also going to add 1/4 of a teaspoon of salt. Excuse me. Ooh, it's a little bit much. I'm going to dump some of it back into the sink. Okay. And I'm going to add my soap. All right, I'm just going to use one of my spoons to kind of stir this up, making sure that all of that soap is mixed in, but also that the salt is dissolved. Okay. Looks pretty good to me. All right. Now what I'm going to do is I'm going to take my plastic bag and I am going to add a strawberry to it. Get this out of my way. All right, I'm gonna take strawberry, place it into my bag. I'm gonna pour my extraction buffer into the bag. And I'm going to start to seal it up, but here's the trick. You want to get rid of any extra air that's in there. So I kind of just squeeze it as I'm continuing to shut the bag. Or if you're very careful, you can kind of lay it down and make sure we push all of that air out. All right, making sure that it's closed. All right, now here comes the fun part. We've got to mash up the strawberry. So very carefully and hopefully not breaking the bag, we want to squish this up. Now, if you do break the bag, it's not the worst thing in the world. It happens. It happens to me sometimes. You're just going to need to get another bag. All right. So I'm carefully doing this. It takes a little bit of time. But while I'm doing this, let's talk about why I just put soap in there. So Mr. Cole does a really great job of explaining what the soap is doing on a chemistry level. But what we're doing here is we're basically trying to dissolve or break down the membranes of the cells, okay? So we have the cell membrane on the very outside, and then remember we said that the DNA is specifically kept in the nucleus, so we're also trying to break down the nuclear membrane as well. The reason why soap does this for us, I'm just going to put this down for a moment. The reason why soap does this for us is because its job is to basically break down or dissolve fats, okay? Think about if you were to have a greasy pot or pan, you were to run it under the sink with just water. Is that going to do the job of cleaning that pot or pan? No, that grease is probably going to stay on there pretty well. But when you add soap, it's able to wash it away. And the reason why it's able to wash it away is because the soap is breaking down the fats, which is the grease in that case, breaking down the fats or dissolving them and making it easier for it to be washed away. In this case, the cell membrane and nuclear membrane are made up of lipids, which are basically fats. So we're helping break down and dissolve those outer layers so that we can get to the DNA inside of those cells. All right, so I think this is pretty mixed up in here. It looks good. We still see some chunks, but that's okay. We still see the stems in there. That's fine as well. So what we're going to do in this next step is we are going to strain it, okay? So I have a cup here, and I'm going to strain it using cheesecloth because I just want to get the liquid that's in this bag. I don't want to get any of the chunks of strawberries. I don't want to get the stems. I don't want to get the seeds. So we're just pouring out that liquid. So I'm just going to take a scissor. I'm going to very carefully cut just a corner out of my Ziploc bag. Just a small one. We don't want too much pouring out. And I'm going to carefully pour it onto my cheesecloth. Now this is much easier if you have a partner with you to hold the cheesecloth in place. But as long as we're just blocking any of those chunks from going in there. Okay, it just takes a moment for it to go through, but we actually don't really need too much of this liquid. But I'm sure you're noticing as I'm pouring, we're seeing that mixture come down to the bottom. All right, I'm gonna pour just a little bit more. Okay, I'm just gonna set this to the side and my cheesecloth to the side. All right, so now I've got just a nice liquidy mixture here of broken down strawberry cells. All right, now for you to be able to see this clearly, I'm going to transfer it into one of these tubes. You don't have to do that, don't worry about that part. But I just think this will make it a little bit easier for you to see it on camera. So I'm gonna take this and pour it in here. Just holding it over the sink for a moment so that I'm not spilling too much. All right, so that'll make it a little bit easier for you to see. Okay, excellent. So right now, and I'm gonna bring you a little bit closer. Right now what's in this tube is DNA, strawberry DNA. Can you see it? No, we can't see it right now. Where do we think it is? Is it at the top? Is it at the bottom? Where? It's everywhere, okay? It's everywhere within this tube. But what we're going to do in our next step is we are going to bring all of the DNA to the very top of this tube. And because it's all clumped together, we're going to be able to see it with our own two eyes. All right, now I'm gonna see if my lab partner wants to join us for a moment because he is what I use every day in my classes. I give the example of him. All right, so this is Chief. And Chief is going to be my lab partner for today. And Chief sheds a lot, okay? So I'm gonna relate this to the DNA. Chief sheds a lot. It's crazy. Good thing my camera isn't good quality right now because you would be able to see all the dog hair on my shirt. But when I walk into my house, I don't necessarily notice the dog hair on the floor right away, all right? But if I were to sweep the whole room and bring it to one spot right in the middle, would I then notice the dog hair? Yeah, absolutely, because now it's all in one place and it's all clumped together. It's the same thing with our DNA right now. The DNA is all throughout this tube. We want to bring the DNA all up to the top, and because it's all at the top in one spot, we'll then be able to see it with our own two eyes. All right, sound good? All right, I'm gonna dismiss my partner right now. He can now go off, and I'm going to get my last part of our experiment. So I have been chilling ethanol in the freezer. And this is just ethanol that you can find. It's rubbing alcohol. You could probably find it in your medicine cabinet. It smells like a doctor's office. Remember, in science, we never just sniff anything. We don't put our nose directly into anything. We waft it. And this is that same liquid they put on your arm before they give you a shot. It feels cold to the touch. So it's the same thing. And what we want to do is we want to layer our ethanol on top of this strawberry mixture. Okay, the way I do that is I'm going to hold this on an angle and I'm going to slowly add it. If some drips out, that's okay. It does dry up very quickly, but I'm going to just slowly add it on an angle because I want that ethanol to be separately on top. Okay, looking good so far. All right. We take a look. Hopefully you can see there. You seeing those bubbles there? Where they look like bubbles? That's actually DNA right there. Okay, so now we're finally able to see it with our own two eyes, which is really awesome because it's being all clumped up at the top. So I'm going to take this DNA out just so you could take a look at it. I'm going to be using a plastic loop. We have these at the lab. That's going to help me, but you can use a toothpick. If you have droppers, you could always just pull out some of the ethanol that has the DNA in it, whatever's going to be easier for you. So I just carefully take my loop and go around the edges, and I can just pull some up and out. And if you could see that little bit on the end there, that is DNA. All right, awesome. Now, if you want to keep this, you can. If you have a little tube that it can go into, just put a little bit of ethanol in the tube and then you can add it to that as well. All right, so hopefully you'll get a chance with some of your items at home to try this experiment on your own. Take a picture of your results, send it to us. We're on Facebook, Instagram, Twitter, and a really great experiment to try on your own would be maybe to try this with different fruits and see if you get more or less DNA, or if you need to change the way the soap buffer is. If you think back to Mr. Paul's experiments, each time he did it, it was a little bit different of ingredients he had to use between extracting it from human cells to when he extracted it from wheat germ. We've now extracted from strawberries. Let's see what else you're able to extract from. Thanks so much for joining us. Have a great day.